This is a description, graphic description of cancer as we live it, as patients, as oncologists, and as we should relate to it as biologists. Tumor is diagnosed, it's removed, typically through surgery, and then an anxious wait time starts for many patients. A time that can be months, years, decades, even, until metastasis may emerge. And if metastasis emerges, that is harder to treat than the primary tumor. In some cases, time is so short that metastasis is already present at time of primary tumor diagnosis. We call that a stage 4. As cancer biologists, we know a lot about the steps that lead from one end to the other of this process. Dissemination in form of single cells and clusters involving a number of molecular and cell biological steps and events, extravasation, installation of these cells as seeds in distant organs, multiple organs, all organs, really. And then, after that time, an outbreak may result if these cells were not eliminated. And the outbreak becomes very heterogeneous, can easily become drug-resistant, and it will be taking different shapes, as we've seen through the meeting, depending on the organ site. Now, we are coming out of an era where cancer research has been dominated, not exclusively, but a mighty presence in it has been oncogenomics, mutations that provided the answer to many of the questions related to how cancer occurs, how does it emerge. We've seen thousands, literally, of measured papers and advances on that. But a complex process like metastasis with so many distinct biological steps, it seemed unlikely to us and many others that it would be explained by metastasis causing mutations. Indeed, there are many examples of searches that have been a dry well with regards to identifying genes whose mutation makes a cancer cell highly metastatic. This is just from one example with my colleagues, Atzlo and Kettering, mining on the massive oncogenomic analysis of patients, Atzlo and Kettering, 25,000 patients, close to 100,000 different tumors. It's a tremendous resource, very highly annotated, very important in its usefulness for a number of studies. But one upshot from it is that not a single metastasis causing mutation could be identified through this process. And this is a repeated occurrence. Now, we now know, and this audience knows very well, that metastasis is a deal between cancer cells and an ecosystem. It's a highly inefficient process. There is massive attrition at each one of those steps. It requires tremendous adaptation of these cancer cells, leaving a tumor, to different ecosystems, different tissues. Each of these tissues provides barriers as well as opportunities. Among the most mighty barriers are immune components, innate immunity, resident innate immunity as well as adaptive, and other barriers, blood-brain barrier, and so many more. Metastasis is enabled by the phenotypic plasticity of stem-like cancer cells. We are now arriving with confidence at this concept. And Cedric was just one more example of how the ability to adopt phenotypic states that are supportive is key for the cells that act as metastasis-initiating cells. We should not lose sight of the fact that metastasis and its ecosystem is not limited to just the tumor microenvironment that surrounds it in the organ of residence, of emergence or residence. Rather, there are now increasing lines of evidence that many systemic components and factors, immunity, endocrine, nerve, innervation, stress, and others that have been mentioned already during the meeting, impact on the progression and response to treatment of the tumor. So this is the nature of the problem that we are all confronted with making progress on for the benefit of patients. How do we tackle this complexity? You have to start somewhere. And where we started, my colleagues and I, many years ago, was to leverage the organ-specific nature of the metastatic spread from, in certain cancers, breast cancer, lung adenocarcinoma, to begin to scrutinize what may be important as epigenetic events leading to expression of specific genes or gene programs for metastatic colonization, overt metastatic colonization of these distant organs. The basic approach and idea was to posit that in a patient with multi-organ metastases, their circulating cancer cells accumulating in the pleural fluid in this case might represent a demographic cross-sections of all the metastatic species that had evolved in that patient, and that a mouse could do us the favor of acting as a cell sorter to separate these cells into brain metastatic, lung metastatic, bone metastatic, and so forth. And then by a scrutiny of these cells through gene expression profiling in those days, one might identify genes associated with a mediating metastasis to that site. Genes whose expression in primary tumors of patients indeed was associated, correlated, predicted metastasis to that particular site. That was a good guess. It was productive. It identified genes mediating extravasation or seeding, initial survival, or overt colonization by breast and lung cancers to a number of important organs. Each one of these genes was surrounded by fascinating biology that was described in individual papers by a fantastic group of colleagues that I had the fortune to attract to the lab and do all this. 90% of them now are running their own independent laboratories pursuing this work. Important as this is, and important as it is, as we've heard in several talks, to identify organ-specific metastasis mediators and therapeutic strategies that might be developed to address that, there is a level of insatisfaction, a level of concern about this approach as the only approach. And that has to do with the fact that many patients are at high risk of metastasis to multiple sites, or have metastasis in multiple sites. We've learned from the case of HER2 breast cancer that treatment with trastuzumab was started by being very efficient against visceral metastases, but in those patients whose visceral metastases were very effectively controlled, it was only to see that within a few years they began to come down with brain metastasis. That now can be treated, but this is a spectacular example how intervention changed the metastatic pattern of the disease. So because of that, we began to turn our attention to a search for common mediators, metastasis mediators that are important for multiple organs and perhaps for multiple cancers. The first one that emerged, and the one that emerged most mightily, was is a cell adhesion molecule, L1-CAM. The way we ran into L1-CAM was by actual observation of what metastatic cells do as they extravasate as models that we have developed, and they are inoculated into the bloodstream of the mouse, let them do what they can, and they will extravasate if they are BRM, brain metastatic cells, in the brain preferentially. If we track these cells by imaging of ex vivo brain slices taken from mice that contain in their brain cells that were in the process of extravasating, they do what you see here. They extravasate and they begin to crawl up and down capillaries of the brain, pushing out of the way these green pericytes. And as they do that, they eventually stop, they arrest, and they extend themselves on the capillary, and that is something that we see precedes division, proliferation. As they proliferate, they form a sheath around the capillary. They don't form an spheroid in most cases of, that we have examined. This is lung adenocarcinoma, same for kidney, renal carcinoma, same for triple-negative breast cancer. We can see these with now improved light sheet microscopy and clear tissue in the whole brain level to see that at any point in time there are cells that are aligning themselves in pink on the green capillaries. Some of them are still single cells that either extravasated later or they may be quiescent, dormant. Others have begun to proliferate, forming a sheath that engulfs an entire local network of capillaries. And as they do that, they begin to attract the attention of reactive microglia in cyan color here to begin to deal with the microenvironment, evidently in a way that they profit from. We identified the molecule that mediates the cell adhesion to the perivascular basal lamina. It was L1-CAM. L1-CAM, known to collaborate with integrins, which we verified in our case, to signal to the cells. And the signaling that we observe by L1-CAM in this context of the cancer cells adhering to laminin in the perivascular basal lamina is through ILK and PAC activation of YAP and MRTF as a signal that is all right to start proliferating after having come out of the vasculature through an ENT and reverting now to an MET. What was interesting for us and encouraging for us is that this, which was initially observed with lung adenocarcinoma and triple-negative breast cancer in the brain, applies to these cancers in other sites and to other cancers as well. So we had the first case in the lab of a mediator of metastasis that is common to most sites. This is some of the biochemistry that was done by Emra Er to define how it is that L1-CAM is important for this process. Neglected to say what the image was showing, which is if we knock out or knock down L1-CAM in these cells, the cells can still migrate, extravasate, adhere to the capillaries, integrins within, but they do not spread anymore. And with that, they fail to proliferate and form metastasis. So it's a mediator of metastasis. It signals in this context through ILK, as I said, to YAP and MRTF, mechanotransduction transcription factor activation for proliferation of the cells. L1-CAM also establishes homophilic interactions at cell-cell boundaries. We are now finding, in work that I will not be able to present today, hopefully next time, we are finding that this interaction drawn here in the cartoon is also extremely important and is signaling in a manner that is quite different from the L1-CAM basal lamina interaction. Now, where do these L1-CAM positive cells emerge? Where and when? Evidently, if L1-CAM is necessary for metastasis, they must arrive with it, they must develop in the primary tumor. Indeed, there is an extensive literature by many colleagues who studied L1-CAM and cancer before we did, identifying expression of L1-CAM, ectopic expression as they called it, because L1-CAM normally is expressed in the CNS and in the neural cells of the CNS. So ectopic expression, as they called it, was associated with poor outcome in many different types of cancers. Nobody knew why. It would probably have something to do with metastasis. Now we know it does. However, as seen here in normal tissue from the intestinal mucosa, human-derived, in studies championed by Karuna Ganesh in the lab, L1-CAM is not expressed in normal tissue. In intact tissue, it's not expressed. Very rarely seen there. However, in primary tumors, as the tumor grows and becomes invasive, L1-CAM expressing cells are seen in the invasion front. In metastases, there is even a higher concentration of these L1-CAM expressing cells, especially if the metastases are still in early stages. Moreover, in samples from patients treated in the neoadjuvant setting with chemotherapy, with an elimination of tumor mass from primary tumors or metastases, the residue, is highly enriched for L1-CAM. Karuna went on to ask, functionally, what might L1-CAM be good for these cells? And some experiments indicated that L1-CAM is not needed for the emergence of the tumor, the adenoma state, at least. However, when she prepared organoids from patient-derived tumor samples, metastasis samples or primary tumor samples, And she sorted the L1-CAM high from the L1-CAM low cells. She found that the L1-CAM high cells are the ones that have the capacity to regenerate organoid and the capacity to regenerate tumor implanted into mice, either subcutaneously or orthotopically or via the bloodstream or intrasplenic injection to form metastatic colonization. Because of its association with the ability to regenerate the tumor, we began to suspect that L1-CAM might normally have something to do with regeneration. So this is why this experiment was done. This is in normal mice with conditional allele of L1-CAM. This is not tumor biology. This is just normal intestinal homeostatic and regenerative biology. The finding was that the knockout of L1-CAM in the intestine doesn't affect the mouse viability at all. However, if the intestine is ulcerated with ESS, now these mice will not be able to heal the wound and they will die of dehydration and diarrhea. L1-CAM that is not expressed in the normal intestinal epithelium of the mouse, as in the human, becomes very strongly expressed in these regenerative progenitors that are healing the ulceration, the wound. Giving rise to this cartoon at the bottom, summarizing this cartoon at the bottom, which says that in normal regeneration, cells emerge, are mobilized as regenerative progenitors that express L1-CAM, are marked by it, and need it in order to do their job of reepithelializing the tumor. That began to support the idea that, first of all, homeostasis and regeneration are driven by different types of stem cells. That's something that resonated with many findings of others who studied regeneration elsewhere. That the L1-CAM expression is important, at least in the intestinal epithelium, for this process, and that this is recapitulated in tumorigenesis. The tumor is initiated from tumor-initiating cells or cancerous stem cells, LGR5 positive, extensively studied by many. That is not regenerative biology, that is homeostatic biology with mutations in the Wnt pathway, etc. But as the tumor forms and begins to injure itself, now this gives rise to, through mobilization of the appropriate malignant progenitors, to the regenerative progenitor state, L1-CAM, and this is what is best endowed to initiate tumor regeneration at a distance. In contemporaneous studies in the lab, the notion that metastasis has a lot to do with normal regeneration adopted by cancer cells, which are regenerative progenitors that happen to be malignant. That additional insight came from studies being done by Ashley Loughney, which was applying single-cell analytics to human metastasis samples from lung adenocarcinoma. This is the spread of the entire tumor and its microenvironment, making points that are important but already known to you, which include that the epithelial carcinoma component is only a modest fraction of the total tumor. The majority of the tumor is inflammatory and immune cells, also niche cells, endothelial, mesenchymal, and so forth. The real insight relevant here is that when Ashley ordered the single-cell transcriptomes according to a lung epithelium development regenerative signature derived by others, Krasno and other towering figures in that field, nothing to do with cancer, normal developmental regenerative biology. The metastatic cells ordered themselves, now just the epithelial component, ordered themselves beautifully along a path that covered one after the other all the markers, normal markers, transcriptional markers of the various developmental stages. From the very primitive endoderm marker to to SOX2, a marker of early-stage progenitors in lung regeneration as well as formation, and the rest of the components of the path towards more extensive differentiation, SOX9 representing progenitors, but more advanced, more differentiated than SOX2. So that told us several things. First of all, that indeed metastasis smells of regeneration co-opted by metastasis-initiating cells. But second, that tumor heterogeneity, which is frequently thought of as something coming from Mars, we don't know where it comes from. In fact, a lot of the heterogeneity has to do with the fact that a tumor, certainly the metastasis, is a flow of phenotypes that are emerging from the pool of stem cells that are contained in there. Third, together with this heterogeneity, there is heterogeneity in other things that are of interest and will be relevant in a moment. For example, heterogeneity in immune susceptibility genes and processes. We take now the same cells, order them in the same way based on this signature. These are the SOX2 positive and the SOX9 positive, early stage, later stage progenitors. And now we are querying MHC class I molecules as determinants of recognition by the adaptive immune system. The series of ligands for NKG2D, the receptor in natural killer cells and CD8 cells. And you can see how there is great heterogeneity associated with the developmental stages of these cells in the metastatic lesion. How these cells would be predicted based on this to be more sensitive than these ones to NK cell killing and CD8 killing. How these ones might be more susceptible to adaptive immunity killing. So with this, we have an example of a class of molecules and a biological process that is worth thinking of targeting because sits at the root of the initiation of metastasis, at least in lung adenocarcinoma and in colorectal cancer. With L1CAM as an example, we are encouraged that this is feasible, that we don't have to restrict ourselves to trying to do something about metastasis when it's already overt, for which we do have therapy, immunotherapy and personalized medicine. A lot of it is being applied to shrink the metastases that patients that we care for have. But in addition to this, making it hard for metastases to initiate or after treatment of metastases with chemotherapy and immunotherapy to reinitiate as a second round of relapse, driven by the same kind of progenitors, most likely. Could we prevent this in the first place? Prevent this by eliminating something that has already been talked about, cells when they are still in the dormant state. I'm going to present to you data summarizing recent findings of the lab, and then more data that is unpublished, about the idea that dormancy is not a static state where all the cells, thousands of them distributed through the organs of the patients, are sleeping, are in quiescence, but they are rather in balance, in equilibrium, with a minority that at any given point in time have re-entered the cell cycle and are being recognized by the immune system and eliminated right there and then. Many years ago, we started to study dormancy by developing models, in this case, not derived from aggressive advanced metastatic state, but rather from early stage, stage 1 and 2A tumors from lung adenocarcinoma patients, as well as from HER2 breast cancer patients. The approach was the same as before, except in this case, we were not looking for overt metastasis. We were looking for no metastasis. It took me a bit of effort to convince the postdoc to initiate the project with this perspective. So cells were injected into mice, allowed to sit without forming metastasis. One mouse in 10 might develop one after several months. And after several months, those organs were harvested put in culture to isolate any cells, cancer cells that might still be present and that could proliferate well, so these were not dead-end senescent cells. Cells that had been sitting in the mouse, able to proliferate, that can be re-injected, and again establish a dormant state, except in one unlucky mouse among 8 or 10 where an aggressive metastasis would emerge, as it happens in people. This is a summary of, after interrogating and studying these cells, of what these cells look like. They are, again, early-stage epithelial progenitors, SOX2 positive in the case of lung adenocarcinoma. They occupy perivascular niches in the brain, lung, kidney, and liver. They are competent to enter a self-driven quiescent state. They produce DKK1, inhibitor of Wnt ligands. Wnt, of course, as you know, are powerful mitogens for stem and progenitor cells. They are highly responsive to TGF-beta that induces deep quiescence in these cells. They are observed as mostly single cells in quiescence, but here and there, about 10% of the population is in the form of small clusters, 15, 20 cells, 4 cell divisions that are positive for Ki67. So they are proliferating. Some of these do manage to form outbreaks, but most of them do not. They disappear, because when we look later, they are gone. That mouse still has only tiny clusters. We also observe that these cells, when they enter quiescence, they downregulate very well multiple ligands for the NKG2D receptor of NK cells. This is an important experiment that Srinivas did in those early studies that has been tremendously influential for the work that we have done since. This is the normal protocol. The mouse contain disseminated dormant metastasis. One mouse among 8 or 10 does develop a metastasis within the span of 3 to 6 months, but most of them not. Now, having seen that there is this downregulation of NK ligands, we ask whether NK cells are the source of trouble for the cells that enter the proliferative state and that upregulate, when they do that, their NK ligands. And indeed, the moment that we depleted NK cells in these mice, and this is an athymic model, so no adaptive immunity, but NK cells were there was metastasis flaring up in all organs and multiple lesions. We tracked later on, actually again, via single-cell analysis, how these dormant cells develop, and indeed, these cells are, when they are dormant and when they are beginning to proliferate, very tiny micrometastases, they are dominated by the SOX2 state, early progenitor. Experiments in vitro and in vivo correlates demonstrated that this state is has the highest susceptibility when it proliferates, not when it's resting, to killing by NK cells. And if these cells get lucky and are able to bypass NK surveillance, then the macrometastasis is dominated by the SOX9 state that is now less susceptible to elimination. We've now reproduced these findings in a syngeneic immune-competent model derived from KP, KRAS, P53 mutant tumors, lung adenocarcinomas in mice. From early-stage tumors, we derive early-stage cells, and these cells have also the capability of establishing themselves as disseminated dormant disease. However, if we deplete NK cells, or now in the syngeneic mice, CD8 cells or CD4 cells, metastasis outbreaks are very significantly accelerated. So that led to the model, or ratified the model, that dormancy is not a sea of quiescent cells that are all quiescent until the day that they wake up, but rather is a balance between largely proliferating progenitors and a few that, for a number of factors, including, I'm sure, some of the ones that you've heard about, are enticed to stop proliferating. When they do that, they do it at great risk, because now they will become immune susceptible. They would be detected by immunity through regulation, upregulation of NK ligands and other things that we want to know about. And that is the question that we address in the study that I will end my presentation with, which is that to ask what cell intrinsic factors lead to the elimination of dormant metastatic cells that re-enter the cell cycle. It's still dormant metastasis as a process, but those cells are proliferating, they are trying, they are going to fail. What makes them fail? I'm not talking about what immune cells attack them. We already saw NK cells, CD4, CD8, you can subtype them. Now, we wanted to know what in these malignant cells makes them susceptible to attack when they proliferate. To address this question, we did a CRISPR screen in vivo with the agency of Francisco Sanchez Rivera, at the time with Scott Lowe. He is now a faculty member at MIT, in studies that in my lab have been led by Jing Hu. So we generated libraries of guide RNAs targeting all the major non-components of immune susceptibility in cells, sick cells, viral infected cells, cancer cells. Introduce that into the dormancy competent cells, and look for guides associated with premature metastatic outbreaks. The pathways or groups, classes of molecules that we targeted are listed at the bottom: NK ligands, MHC class I molecules, DNA sensing STING pathway, RNA and lipopolysaccharide sensing pathways, interferon response, complement pathway. Several of them scored. Some of them were expected. MHC class I molecules, well, that's good to know, but that is not a shock. NK ligands, we already knew that we can deplete NK cells and these metastases will progress because they were being prevented by them. The one that we didn't necessarily predict that would appear is the STING pathway and its client pathway, the interferon pathway. This is some of the data. This is plotting both genes that score, or guys that score against the genes, corresponding genes, both in the human model and in the mouse model of latency. STING itself, CCL5, downstream cytokine, interferon responsive genes associated with it. Indeed, when we look by immunofluorescence, we see that in the population of disseminated cancer cells during dormancy, the single cells that are not proliferating express low levels of STING. The cells that have started to proliferate recently re-entered the cell cycle. They are still in clusters of 8 to 16 cells. They have the highest staining for STING and CCL5 as its downstream correlate. And this is observed both in the human and in the mouse models of dormancy of lung adenocarcinoma. When we look at the level of single-cell analytics in cells that were taken from dormancy and put in culture in vitro to induce a massive entry into the cell cycle, again we see that these cells, which are SOX2 positive, are the ones that show the highest expression of STING. This is corroborated also in patient samples. In micrometastatic disease, in lymph nodes from lung adenocarcinoma patients, single cells that are Ki67 positive have lower levels of STING expression and CCL5 expression than tiny clusters that are Ki67 positive. As the tumor progresses, if it progresses, it's because somehow the cells bypass that. We see that in pair-matched samples, primary tumors and metastases, the metastases that did progress have lower levels of STING, a correlation that fits with the model. Now, there are some cells that are still expressing STING. What are these cells? If we consult the single-cell analytic profiles, indeed, within the metastases, if we look at the progenitor cells, it is the earlier progenitor that live in metastases that have the highest levels of STING, STING pathway components, TBK and IRF3, as well as STING downstream signatures, NF-kappa B, and additional STING targets. Necessity and sufficiency experiments. What happens if you downregulate the STING in dormant cells? The answer is they come out more avidly, better. Metastasis is accelerated because we are removing this liability. They are killed less well. If we overexpress STING in cells that form a spontaneous outbreaks in these mice that have downregulated the STING, now we give them trouble and metastases are slowed. The STING-associated metastatic death of these metastatic cells is indeed associated with infiltration of the small tumor populations or lesions with immune cells, and both NK cells and CD8 cells are necessary for this STING-associated liability, as one might expect, and these results show. So this says that STING expression peaks in dormant metastatic stem cells that are re-entering or upon re-entering the cell cycle, and mediates their demise, mediates their elimination in an immune-mediated manner. We have done analysis of the epigenetic modulators of all these transitions, how in response to TGF-beta, the inducer of dormancy, there is a downregulation of STING expression, and that is associated with loss of activation mark, H3K4 methylation, whereas the selection for cells that can escape and form a breakthrough lesion and reach for SOX9, their methylation of the promoter and the enhancer appears to be the preferred way to silence STING expression. Now, with these, do we have a chance to leverage it in order to get more of what STING does? More elimination of disseminated dormant cells. Fortunately, this experiment was made possible by orally bioavailable agonists of the STING pathway developed by two groups. The one cited here is at Merck. An agonist that was shown to be active in vivo to hyperactivate STING in the context of tumors, although in their case the intention was to hyperactivate the STING in the immune system. So we did the following experiment, Jing-Hu did. Take these mice, load them with cells to establish metastases through the circulation, dormant metastases in multiple organs, and do just 2 rounds, two dosings of the inhibitor, MSA2. And then divide the cohort into 2 groups. One that is examined right away for disseminated tumor cell counts in various organs. The other that is allowed to proceed and form metastases if they can over time. As you can see here, treatment with MSA depletes quite extensively in a short period of time the dormant cell population in these mice. This requires a STING expression in the cancer cells, so if the cancer cells inoculated to these mice are knocked out for a STING, then there is, first of all, a higher accumulation. Second, the STING agonist doesn't do anything at all. Moreover, in the mice that were allowed to proceed, the addition of the treatment with the STING activator very significantly delays and reduces the metastasis-free progression as well as the overall survival. And again, this was dependent on STING expression in the cancer cells. The same experiment with same results in the human dormancy model. STING activation or agonist for a STING leads to a depletion of disseminated disease in the dormant state, at least by killing the cells that are putting themselves at risk by trying to regenerate the tumor. We suspect, given the magnitude of the drop in a short time, that even the lower levels of STING present in the true dormant cells might be activated enough, high enough, by the agonist to eliminate even these ones. Experiments to test these are in progress. So, STING-activating drug can do good here. This is a proof of principle. This needs to be tested, of course, more extensively. This is all, first study. And the consequence of this, of course, is a reduction in the probability that there will be metastatic outbreaks. So returning to the opening challenge picture, we now know that in addition to attacking the overt metastasis with everything we can throw at it, there is hope that we can intervene by preventing metastasis or second relapse of metastasis after treatment of overt metastasis with drugs, personalized precision medicine, immunotherapy, leaves a residue that is very likely re-enriched for this. And this is, again, through maneuvers like acceleration, increase in the mechanisms of clearance of the cells, or blockade of factors like L1CAM, that CD36 you heard about, factors that these cells need to reinitiate, or when they reinitiate growth, to have this growth succeed. And with that, I want to acknowledge the champions, the fantastic group that has made it possible. Thank you all very much for your attention, and have a great ending of a fantastic conference. Thank you.